It's my distinct pleasure to introduce our seminar speaker today, Dr. Ralph Larson from San Francisco State University, Professor Emeritus. A little bit about Ralph's background. He did his bachelor's degree at Occidental College, and from that point then he went to UC Santa Barbara, where he did his master's and his PhD work with Al Ebling. After he finished his PhD, he had a variety of sort of fisheries, research biologist positions, different institutions in New York. He's back in Santa Barbara. In 1980, he started the position at San Francisco State, where he then spent the rest of his career as a professor until he retired in 2006, I think it was. A lot of his research interests throughout his career spanned lots of different areas of fish biology and ecology, studying patterns of community composition, assemblage structure, really doing some really important work looking at competition between different rockfish species, to looking at sort of larval dispersal and vertical distributions in the water column of larvae, to in the more later years getting interested in population genetics and identifying cryptic species and a lot of cool work. Today I was lecturing to the subtitle ecology class about ecology of fishes in kelp forest. And there were 3 different times where I was talking about different aspects of Ralph's work throughout his career. So, it really just, spans a really good diversity of topics, and, it's really a pleasure to have him here to talk with us today. Other sort of highlights, beyond sort of his academic career, he was the president of the Western Society of Naturalists. He served here as the president of the governing board at Moss Landing. He was on the board of directors of a lot of different agencies and conservation groups, and so he's really done a lot over his career. And so it's a great pleasure to have Ralph come speak, and he's going to tell us about roadblocks, detours, and paths not taken. Yeah.
Thanks, Scott.
By the way, we're trying a new lighting scheme today, so we're going to go the lights on and see if we still get recording.
Well, it's great to be back at Moss Landing. I haven't been back at Moss Landing Marine Labs for some period of time. I don't know if I've been back since I retired. When before that, it seems like I was here, 2 or 3 times a semester anyhow, with the Board of Governors meeting and student thesis defenses for when I was on committees for umpteen thousand of Greg Collier's students and so forth. And so I really miss being here. So it's great to be back. In fact, so the reason that I came here, that I am here today, is that I ran into Matt Edwards at the WSN meeting last fall, and very innocently asked him, I asked him how's it going. He said, oh yeah, I'm the visiting faculty member at Moss Landing this year. Oh, that's great, Matt. That's really good. And thinking a little bit, I was listening to these great talks at the WSN meeting, and intellectual stimulation, all that sort of stuff, and so I asked him, well, can I get on a seminar list for Moss Landing Marine Labs? I can drive down and go to a seminar and see some people and stuff like that. And Matt's going, you want to come down and give a seminar next spring? And I thought, let me think about it for a minute, Matt. I've been retired for 8 years, and so, I could talk about, I could give you a nice travelogue, here's where we're in Andalusia, and here's where we're there. I could talk about conjugation of Spanish verbs and stuff like that, but I haven't done any new research over the last while. So I thought, well, I'm not going to just give a talk about one old research program that I was involved in or something. That'd be lame, Here's a program that I started in 1982, although I'm going to tell you about one of those today. So what I decided to do was kind of talk about places where I was proven wrong on something, often by Moss Landing students, to tell you the truth. Things where I screwed up, and other things where maybe I wish I could have done a little bit more on. Hence, 4 tales of unfinished business. I didn't really know what to call it, but that's tales of unfinished business. So I'm going to talk about some of my failures, but wind up with each of them, hopefully with something that might, be interesting to somebody to follow up on at some point. So that's what I'm going to do today. My first little tale is Dead Wrong in Public number 1. an instance of being proven wrong about something that I thought I really understood, or I thought I really understood. And that is the settlement behavior. Some of these are like really narrow topics, but you know, I can't just address the huge breadth of my failures in my life, you know, so just little pieces, you know, little pieces at a time. So settlement behavior of black and yellow rockfish, this is one thing. When I was at UC Santa Barbara doing graduate school, I studied species interactions between black and yellow and gopher rockfish. And they tend to segregate by depth with black and yellow rockfish occurring in shallow water and gopher rockfish occurring in deeper water. And I was trying to study that interaction between those two species. That was my doctoral dissertation. And these counts are from some study sites I'd set up out at Santa Cruz Island. Well, when I did these counts, how do I do this? Okay, this one. When I did these counts, I kept track of the size of the fish. You know, I had size categories and stuff, including young of the year fish. And only 1 year of the three summers I was in the middle of doing this project did I actually see a recruitment of very many of these fish. And so I counted them, and it turned out that lo and behold, the black and yellow rockfish the young ones that I saw tended to be in shallow water, just like the adults are, and the young of the year gopher rockfish tended to be in deeper water, just like the adults were. And I took that to be sort of an important, what I figured was an important part of the process of involving the segregation of these very closely related species, their segregation and ecological differentiation. That's how the distributions got set up. And that's still true. That's still true. I wasn't wrong about that. But what I was wrong about was the method of settlement that gets into that point. What I thought was this. This is the hypothesis that I thought of. And it seemed reasonable to me. And that is that black and yellow, I thought that maybe at the end of their pelagic stage, juvenile black and yellow rockfish would be up high in the water column. Maybe they're up in the Newston or something like that. And they drift into the kelp, drift into shore, and they wind up in shallow water. Makes sense, right? And then the gopher rockfish juveniles, when they're ready to settle, maybe they're orienting below the thermocline or something like that that gets them into deeper water. And so when they encounter shore, they're in deeper water too. So I thought that would be the way it happened. You know, I thought that was a clever idea. And part of the reason I thought of that was that there was an offshore reef off north of Goleta, actually, called Naples Reef. And it doesn't come up to the surface. And at that point, at that spot, I documented it very well. And in some other places that are offshore reefs, it seemed like black and yellow rockfish weren't particularly common, and gopher rockfish occurred in relatively shallow water. And so these are counts along some transect lines I put out there, and these are the proportion of black and yellow rockfish out of the two species. And here's a transect line in 20 feet of water where black and yellows on average made up less than half of the fish that I saw. Interesting observation. You make observations, develop a hypothesis to go for it. My hypothesis was that, this is actually sort of the genesis of what I just showed you a minute ago, is that if the black and yellows are up high in the water column, maybe they just drift right by the top of the reef when it's time to settle and they miss it. And that's why you don't see them. But then the gophers are down there below the thermocline or wherever they are. They encounter the reef, and so that's why you see so few black and yellows and so many gophers down there. That was my story. I think I actually wrote that part of it up in my papers and stuff. I don't really remember anymore. But then I came to San Francisco State in 1980, and at that time there was a group of students here, master's students of Calliers, Mark Carr, Todd Anderson, Mickey Singer, and Guy Holzer. You may have heard of a couple of these people, perhaps. I don't know. But they were studying rockfish recruitment in the Monterey area. It was sort of Milton Love's idea. They took it, and they basically invented the study of fish recruitment to the nearshore in carrying out this study that they did in Monterey on rockfish. And I arrived and having done all this, some work on rockfish, and Mark, and I can't remember, I know I was on Mark's thesis committee and Todd's thesis committee, and I can't remember about the others. But anyhow, I talked with them and everything like that. And they did a really good job. It was great work. But they proved me dead wrong in public about the settlement behavior of black and yellow and gopher rockfish. What they found, and I remember very clearly having a phone conversation with Mark Carr one day, and he's talking about what he found and everything like that, going it through very carefully, and me saying to him, that is good evidence, and that's all that matters, And that's the progress of science and so forth, although I felt a little silly about it. But anyhow, what they found is that basically black and yellows and gophers, also kelp rockfish and copper rockfish, which all are a form of monophyletic clade within the rockfishes, have a similar behavior of settling in the kelp canopy. Since it's been discovered that all of them are up high in the water column before they settle. They have a number of other similar early life history characteristics in common. So they settle in the kelp canopy, and then as they grow, they spend a little bit of time in the kelp canopy, and then they swim, they kind of work their way down the kelp stipes to the bottom, and then start their life living on the bottom, and then presumably the black and yellows sort of scatter out, stay where they are, they scatter out into shallower water, and the gophers move out into deeper water. And that's the truth. That's really what happens. And so I was wrong about this. I don't remember if I was upset about it. I don't think I was, but I couldn't believe that I'd missed this whole thing. That I must have made like 1,000 dives in kelp forest by then and stuff. Never had a clue. Now when I go out, it's just totally obvious. There they are. So anyhow, I was wrong. It gives the same result, but with a different mechanism. It still doesn't explain why black and yellow rockfish aren't common on offshore reefs like Naples Reef, but maybe that's for somebody else to figure out. But it's a different mechanism, but it's an interesting one, and I think it's more interesting than the one I had, because it really ties into the evolution and ecology of this clade of rockfishes. Here's the clade. That's from Hayden Vetter's rockfish phylogeny paper. Sister species, Carnatus and Chrysomellus. Atrovirens, the next one, and Carinus. I don't know how the tiger rockfish, and down here is the China rockfish. I don't know about their settlement behavior, but it's common in these species, and it's an interesting thing in that the ecological differences between these species really are initiated by what they do after they leave the kelp canopy and come down to the bottom. It's their habitat selection behavior at that stage in their lives which basically initiates the differences in their ecology. The kelp rockfish stay in the kelp. Copper rockfish move out into low relief rock and sand rock interface, and the coppers presumably, gophers move into deeper water, and black and yellow stay where they are. Interesting kind of thing, and maybe involved in their evolution as well. So what I'm thinking about now, and this is based, I've actually thought about this more in the last few weeks when I'm putting this talk together, is that maybe this kind of switch in behavior has been really important in the evolution of these different species, and particularly perhaps between black and yellow and gopher rockfish. Another great thing that happened as I was preparing for this is I discovered a 2011 paper on genetics of gophers and black and yellows that I hadn't been aware of before. That's just totally interesting about these things. I guess they are species, although when I was just finishing this up, I used to get in trouble because people would say, how do you know they're different species? And I didn't, it just seemed like it. In any case, wondering the degree to which this settlement behavior could play a role in their speciation, perhaps not even in speciation that comes through separation of initial populations where they can accumulate differences without gene flow and you come back together again, but whether it might actually be sympatric speciation where there's actually never any geographic separation between the populations. That's an interesting question. So anyhow, I'm wondering about this this is my lesson to come from my mistake, that black and yellow gopher rockfish are one of a number of species pairs of rockfish that segregate by depth. There are a bunch of them, including one pair that was just recently discovered on the basis of genetic evidence. John Hyde, Russ Vedder, and their colleagues actually discovered a cryptic species of rockfish, Sebastes crocotalis, when everybody thought it was all Sebastes mystinus before. They discovered this genetically, they've identified it morphologically, and they came up with a simple hypothesis for how this kind of speciation might occur. And the hypothesis is this, and that is that many species of rockfish tend to settle in relatively shallow water for whatever reason. And then they undergo ontogenetic migrations out into the areas where they, into deeper waters where they spend their adult life. And what they propose is that maybe the speciation mechanism for crocotalis and miniatus is that there was once an ancestral species, kind of like crocotalis, that moved out into deep water, but then in the process of selection maybe for life in shallow water and so forth, there began to be an evolution of a shallow water species, and the part that would separate the adults so that they don't interbreed with them is that it's a simple thing where the ontogenetic migration of these juveniles is just curtailed. And they talked about it as a kind of pedomorphosis, a loss of, or keeping larval or juvenile characteristics into later stages. It's an interesting idea, and I wonder if something like that could have been going on with black and yellow rockfish. That the mechanism for speciation in them, which apparently happened something about 400,000 years ago, could have involved this kind of behavior. So I don't know exactly what you would do to study that process of movement to help resolve the issue. But I think there are a number of interesting things to study about these things. One of them would be to look at, to actually document the movement of black and yellows a little bit better. I don't know how much it's been followed up on, but actually Todd Anderson, Mark Carr, and those guys actually didn't find that many little black and yellows up in the canopy, which is probably reasonable because black and yellows have a much smaller population, narrower depth range, smaller population than gopher rockfish do. But actually document that movement a little bit more. And I don't know if anybody's ever actually tagged juvenile gopher rockfish and documented an offshore movement. I think that would be an interesting natural history thing to do. And then there's a lot more about the genetics of gophers and black and yellows that I think would be interesting to follow up, including examining hybridization, which actually does happen to a small degree. And one thing that these Vince Bonacorsi and his colleagues found that it seems like there's still really intense natural selection going on, particularly in black and yellow rockfish. Figure out what all that's about. So anyhow, that's my first tale. On the topic of sympatric speciation, does anybody know about this? The old conflict and ideas about allopatric versus sympatric speciation. Most popularly, never mind. Well, a former Moss Landing student named Karen Crow. She was actually, wound up being her thesis supervisor. She's now teaching the ichthyology class at San Francisco State, but she published a very important paper on sympatric speciation in greenlings 5 years ago. And I wish you could interbreed rockfish like you can greenlings, but you can't do it. Anyhow, so that's my first tale. The second tale is also dead wrong in public, proven wrong by a Moss Landing student again. Only it's an interesting thing in that it's actually the possibility of two mutually exclusive truths going on. And I'd like to figure out how to resolve it. It has to do with movements of black and yellow rockfish. So when I was at UC Santa Barbara, it came to be obvious to me as I was working on these species that the fish stay put. As I was working my way counting fish along these 30-meter-long transect lines at Santa Cruz Island. I knew that if I looked around the corner underneath this rock, I was going to find a rockfish, because it's always there. And then I saw the fish, having these agonistic encounters and stuff, and I wondered whether this territorial behavior would be a mechanism of competition between the two species. And so I mapped out 3 study sites, tagged about 100 fish, and followed them for a course of a year. Bone-chilling, but it's probably the most fun research project I've ever done. It was just great getting to know all these little guys and stuff. And so, it did turn out that they have really fairly restricted home ranges. These are home ranges of a couple of different gopher rockfish at Naples Reef, about 25 feet of water. These are just different fish, different places that, because they're above one another, doesn't mean anything. But typically, that I would find for a fish that they'd have a shelter hole that they spent most of their time in during the day, unless there wasn't very much wave surge. And then there would be a bunch of places, and that's where they'd be found. These little lollipop diagrams are the number of times I saw the fish in a particular quarter-meter square that I imagined on my map. And then the fish would move around their home range. Actually, they'd go at night. They'd go and sit somewhere else in the home range, maybe. Sometimes there were favorite spots like this one over here, this fish. Sometimes they'd be, they'd like have spots along a ridge like that where they could, touch both bottom and side to things around them. And they defended. These little X's marked sites where I saw the fish defending their territory. And so they'd have agonistic counters, out to the edge of their home range. So I thought they were territorial. And in fact, they were. And they have these really complex societies of rockfish. This doesn't really mean much for the purpose of the talk, but these are the home ranges of a bunch of black and yellow rockfish in about a quarter of my study area at Santa Cruz Island. They're really packed in, and they do all sorts of other interesting things with regard to that. So I finished all that, and my conclusion, if you want to ask me about it, and if you want to ask probably most anybody about it who these days, they would say, yeah, these rockfish stay put. They don't move. I had about 100 fish. I recognized about 100 fish out there. I lost track of 2 of them in a course of a year, and that's easily accounted for by natural mortality. So they don't move, right? They don't move. So again, shortly after I came to San Francisco State, a couple of Moss Landing students, Feeny Matthews and Al Solonsky, had gotten in on a grant to study the development of the fish community and the fishing on a new artificial reef out by Capitola. They're building a new freeway somewhere, and they had a bunch of broken up concrete, so they dumped it out there. Where it's a really short trip for the people fishing from skiffs that they can rent at Capitola Pier and stuff. And so they got a grant to study this. This new artificial reef, and I had just come to San Francisco State, and I was working on rockfish, and Feeny was a student that was enrolled through San Francisco State, so I wound up being on her thesis committee, and so they came up to San Francisco State, and they talked to me and stuff, and Feeny said what she wanted to do was to tag a bunch of rockfish on the natural reefs around there, and see if they move onto the artificial reef. And I said, pretty unequivocally, I think, that it ain't going to happen. I mean, and it still blows my mind about that anyhow. But here's this little artificial reef. It's like several hundred yards over sand bottom from the nearest natural reefs. And I just could not imagine a black and yellow rockfish or a gopher rockfish swimming out over the sand, points unknown. They don't know there's any reef out there, and wind up on the artificial reef. Feeny, you're wasting your time. Don't even bother with this. Well, I meant it as a warning. She took it as a challenge, I think. And so she did it, and guess what? They moved. Flabbergasted, but she tagged 16 rockfish on natural reefs. A variety of these natural reefs around there. And a quarter of them were recovered on the artificial reef. if you extrapolate that, a quarter of the fish were moving from the natural reefs out to the artificial reef. Those are black and yellows. You know, a twelfth of the gophers, or an eighth of the gophers, or gophers did that. I couldn't imagine brown rockfish doing it, but they did it. All these fish moved out to the artificial reefs, and so, well, that was pretty interesting. Well, Feeney and Al actually they published this work, and they drew some interesting conclusions about artificial reefs that actually sort of ruffled the feathers, and I think that's something that Feeney truly enjoyed doing. But the biologists who were devoted to the idea of artificial reefs and how great artificial reefs are and everything, but they basically argued through their work that maybe artificial reefs, instead of increasing fish productivity in an area, actually decrease it by acting as a sink, because you put out an artificial reef, which is the best habitat out there, great vertical relief, lots of holes, lots of surface area, everything like that. And if any fish in the area encounters it, they're going to say, I'm going to live here. So they live there, but then there's a buoy up on top where their coordinates line up for it. So all the fishermen come out and fish on there every day. And so the fish sort of they would have been, maybe nice and happy, live a long life on the natural reef, but they're attracted to this reef where they're pulled out by the fishermen. That was a significant argument that they made. But as far as movement goes, this is the thing about two mutually exclusive truths. My fish didn't move. I watched fish for a year there. They didn't move. I did another tagging study at black and yellow rockfish at Hopkins Marine Station. That went for over a year. Those fish didn't move. And even some of my other graduate students later on utilized those fish. And then Tom Leidig and I went diving. Actually, during a WSN meeting one day, decided to go diving at Hopkins Marine Station. We went out there. We were at one of my tagging sites, and we saw one of my tagged fish there. And it had to be in the same site, and it had to be out there for at least 7 years. So, one truth, they don't move. But then Feeney's fish did move. And so how do we resolve this? There are a couple of possibilities. And given the interest in marine protected areas and movements of fish in and out of marine protected areas and just understanding natural history, it'd be interesting to figure that out. Well, what could be the difference? Well, Feeney's pet hypothesis for this is that she had read a bunch of stuff that showed that counts of rockfish decreased in kelp forests in shallow water during the wintertime. And Gilbert van Dijkhuizen found that. He found that he was a Moss Landing student who worked on kelp rockfish down in Carmel Bay. Found in Carmel Bay that the kelp rockfish moved into deeper water in the wintertime when the surge got really strong. So she thought that maybe they would they move away from these areas during the wintertime, and that could account for them finding a way out. Although they started looking, the reef was put in August, and as soon as they started looking at the reef, there were fish out there, so it didn't require wintertime. And some of my fish were in the same places, bridging over winter. I saw them. So I don't know, that could still be a possibility, and it can be checked out. That's be interesting to know about wintertime movements of fish. Another possibility, and I think this is what I came up with, is that the reefs around Capitola, the natural reefs, are all really low, very low relief rock. I mean, there's not a lot of vertical relief, and it's not, optimal habitat for black and yellows and gophers and things like that. And so maybe they exhibit a little bit more repetitive behavior, and they just kind of keep moving around. They're more inclined to keep moving around, and in their movements, maybe somehow they wound up across a quarter mile of sand bottom and found the artificial reef. I don't know. I think it's maybe a little bit too facile an explanation, to just say that that's the truth, but it's something that could be checked out as well. The third thing is just way out there, but I remember reading a paper by Ernie Reese, who was one of those first generation of fish ecologists who were using scuba and stuff. He studied at the University of Hawaii and did a lot of work on butterfly fish. And he mapped out home ranges for butterfly fish and so forth, and found that, they usually have that nice little home, or nice little or big home range, but every once in a while, get a pair that just take off and go swim all over the reef and stuff, and then come back, some like journey of discovery or something like that. And I wonder if fish do that sort of thing from time to time, and if black and yellow rockfish do that too, but I don't know. But in any case, I think maybe more telemetry work on those could resolve that question. So that's the second of my two tales, and my two times I was proven dead wrong in public by Moss Landing students. The third one has to do more with how you should really think through your project before you actually start working on it, which I didn't do in this one case, but I have an excuse for it. And it has to do, these are like little topics, but something that I remember. The difficulty of estimating daily food ration size when fish are eating hard to digest prey, slowly digesting prey. And this goes back, I don't know, Di, were you still around? Were you at Moss Landing when they having the gut shops? Yeah, there were, back in the early 80s, Greg, Kaye, and you know who Greg is, organized these, a couple of sort of workshops they called Gut Shop on analyzing stomach contents in fish and so forth. And part of that was that some of these people who were developing models to estimate based on stomach contents and the evacuation rate of food from the stomachs, how much a fish ate. Interesting thing. I thought that was interesting too. And so I engaged in a project that was supposed to do that, involve that. And what I did in the early 1980s was initiate a project where I wanted to look at the feeding, seasonal variation in feeding, reproduction, fat and liver reserves, and growth in black and yellow rockfish and kelp rockfish. I had two aims in this. One is that my very first student, Patrick Guillemot, did his master's thesis on seasonal changes in fat, mesenteric fat of rockfish. The National Marine Fisheries Service lab, which was over in Tiburon at that time, had a bunch of guts of rockfish, and he went and cut all the fat off these things and weighed it and so forth. And he was wondering if fat was stored up before reproduction, and that fat might have been used for gametogenesis, in which case you would find fat going up first, and then going down as the ovaries and testes got bigger. And naively, that's what I thought was usually the case. Well, he found that instead of fat preceding gonads, that they went up at the same time. They both went up, and these are for rockfish that spawn in the December, January, maybe even November, and so they're yolking up and getting fat in the productive time of the year anyhow, in spring and early fall, and spring, summer, and early fall. So it was more simultaneous, and he thought that fat then was used by these rockfish during the wintertime when there's less food around, and they're using it for survival, and that goes in with the long lives of rockfish. So I thought that was interesting, wanted to follow up on it. And second, it's sort of a silly little thing about why kelp rockfish grow more quickly than black and yellow rockfish. And I thought it was because they ate small little crustaceans like you find in the kelp canopy, which are digested more quickly than crabs, and so that maybe they actually eat more than black and yellows, despite the fact their stomachs are more empty whenever you catch them, but maybe their food turns over more rapidly. So I wanted to do this, and I started off a pilot study. And so in August of 1983, students and I went out and we started doing some collections. And we collect the fish every 6 hours around the clock. It started at noon, 6 p.m., midnight, 6 in the morning, and we save all the gonads, save the guts, and save the heads and all that sort of stuff. I really intended it to be a pilot study to find out some of the variables, how much variation there is. What are we dealing with in here? But it turns out that I started this project right at the end of the 1982-83 El Nino. And the fish were just emaciated. I mean, their livers were like little pieces of leather, and there was no fat, and they were skinny, although we didn't think to start measuring body weight until a year and a half later, unfortunately. But anyhow, they were skinny, and we measured the fat. These are log scales of liver weight and fat weight, and they just didn't have much in the way of livers and fat at that time. And it's not much of a pilot study when you've got data that are really unusual. The 1982-83 El Nino was like 2 or 3 times in a century event or something, So thought, okay, well, we'll do another year. Maybe it'll be a normal year, and maybe we'll be able to compare a normal year with an El Nino year. So take advantage of the opportunity there. And so we went out the next year, and do you folks know what pelagic red crabs are? Pleuroncodes planipes, where they're galatheid crustacean. It looks sort of like a langostino. They're pelagic, but they have claws and they have a little tail. They're usually found off Baja California, but during the El Nino, they got carried north all the way to Monterey, and they got stuck in the middle of Monterey Bay, circulating around for the next year. And every once in a while, the pod, they get drifted over the shore, and you'd see them washed up on the beach. You'd see them crawling over rocks. You'd see them hanging from kelp plants like Christmas tree ornaments and so forth. And the rockfish, being, the good opportunistic feeders that they are, just chowed down on these things. In fact, the pelagic red crabs were fairly large, and we'd spear a fish and look in its mouth, and it would swallow the tail end of this thing, of one of the crabs, as far as it would go, with the abdomen wrapping under, but it couldn't fit it all into its stomach. And so the rest of the crab was sticking out the esophagus into the mouth of these fish. It's a phenomenon that Al Ebling called rockfish gluttony in a different context. And so that was a really different year, and the fish were fat. The liver went down a little bit in the wintertime, maybe associated with reproduction. Fat was just stayed really high. So they were really fat and happy at that time. And this is truly like 1 or 2 times a century event that something like that happens. Well, guess we'll go out and do it another year and see if we can find a normal year. And which we did. And 86, it was a slight El Nino, but it was, we had to stop after that anyhow. We'd killed 1,000 black and yellow and 1,000 kelp rockfish and had that to do. And so we finished so we finished the project. We finished doing the collections and so forth. And we did find some interesting things. Some good things came out of it. I analyzed the data on the fat and liver and had some interesting ideas, interesting findings, and some interesting ideas to go forward from on that I'm not going to talk about. And a bunch of students found thesis projects on it, which is part of my aim in setting up a project like that. And so that part was great. But here we are, 30 years later, something like that, almost 30 years since that project. And we actually have the stomach contents analyzed. That is, Tom Leidig, Todd Hopkins, Jim Couch, went through all of these stomach contents. We have never published this data. And the reason why is that I couldn't figure out a way to compute the daily food ration based on the stomach contents of these fish. And the way you compute the daily food ration, there are a couple of different methods for doing it, but it depends on some different narrow range of models for evacuation of food from the stomach, linear, exponential, or whatever. And so the daily food ration would be computed as a function of the stomach evacuation rate and the average stomach contents during the course of the day. But it depends on stomach evacuation following one of these models, and that they evacuate all the food they eat in a day within the next day. Because basically you're calculating how much food they evacuated within a 24-hour period. So I knew that black and yellow, that crabs weren't evacuated like that. But finally, a student named Todd Hopkins looked at stomach evacuation in black and yellow rockfish. And this is what he found. he found that within 24 hours, that critical period, that a crab wouldn't even start evacuating from the stomach until about a day after it was eaten, because it takes that long for the shell to get softened up enough to fit through the pyloric valve and so on. And then it goes through some kind of evacuation after that. And so this kind of just left me paralyzed for the last 30 years. One sabbatical in 1986, I thought, okay, well, I'm going to figure this out. But then I started working with the National Marine Fisheries Service at that time, got off in different directions. Every once in a while, I'd look in the literature and see if there's a way to figure this out, and I couldn't. So it's just there. Tom Leidick up at the National Marine Fisheries Service looked at 1,000 kelp rockfish stomach contents, and he's not happy about not doing it. So the question is, why didn't I plan ahead and figure out about digestion of crabs before I started the project? And I think I did know, I knew it was going to be not exponential evacuation. And I knew it was going to be a long time, but I think I just thought, we'll figure that out later on. So I didn't do that. So why didn't I plan ahead? We can ask my wife about that. It's fairly simple. However, and I'm very grateful to Matt for inviting me to do this, because it stimulated me to prepare this talk and to actually look at papers citing Todd Hopkins' research on stomach evacuation. And just, this is like within the last couple of weeks, I found a couple of papers published that do come up with a method for estimating daily ration size in these weird kinds of things. And it's sort of what I thought is it's all ad hoc. You have to know the state of digestion and a bunch of different things. So I'm talking with Tom Leidick now, and either we'll figure out a way to use this, or maybe we'll just write to some of these people and say, hey, we've got a great data set to use. You want to analyze it for us? And just stick us on at the end as authors sometime. It doesn't matter to my career anymore, but it could to Tom's. So anyhow, that's the second thing. One third thing. My last thing, I don't really have any regrets about it. I probably made many mistakes, and many more will be discovered. But I think there are just some interesting questions. And during the last 10 years or so, when I was working, a lot of my students, like Lisa Kriegsman right here, looked at something that I was interested in, is geographic variation in population biology, and how do fish populations work at the extremes of their ranges. And students did a bunch of different things. They looked at growth rates. Jody Zeitlin, she was actually one of the people involved in that project in the 80s. She looked at growth and reproduction of black and yellow rockfish in Central and Southern California. A later student named Kevin Walters looked at blacksmith and senioritas, Central and Southern California. And fish grow at different rates, and often what you see is that in further north, fish grow bigger, and they often seem to live a little bit longer. That's one kind of difference. Another difference has to do with recruitment patterns. That is, that how many, in what years do you have lots of baby fish showing up? And Kevin Walters, who studied blacksmith and senioritas, compared the recruitment patterns of these fish between Southern and Central California. And the way we did all these projects, because we couldn't go back and look at recruitment itself, the baby fish, we collected adult fish, determined their ages, and then from that, we'd be able to calculate or estimate what their birth year was. And so what Kevin found for blacksmith, which is a member of a tropical family, it's a damselfish, and occurs sometimes in Monterey area, was that in Southern California, they pretty much recruited, that young appeared all the time. But in Monterey, the fish that in our collections, which were made 1999, a little bit in 2000, most of the fish had settled during the El Nino of 1997 and during the El Nino of 1992 and 1993. And that matches a lot of observations that people have made of these warmer water fish, Southern California fish in Monterey before. Looking at it from the other direction, a student named Paul Gundelfinger looked at blue rockfish, and this was before we figured out that there were 2 species of blue rockfish. But we made collections of blue rockfish in Sonoma County, Monterey, down near San Luis Obispo, and then out at Santa Cruz Island. And this is a different kind of graph. Paul did analyze the data a little bit differently, but the lines above are good better than expected recruitment. The bars below are a little poorer than expected recruitment. And there's a little bit of general pattern in there, in that 1998 showed up to be a bad year in a couple of places. 1999 was a strong La Nina year. It was a cold year, and there was good recruitment in some of those years, and so on. But basically, in Central California, we found that the general pattern was that actually fish grew bigger and lived longer in Sonoma, whether that was type 1 or type 2 or A and B, I don't know, but that actually the diversity of successful year classes decreased as you went south, and significantly so down near the southern end of Central California. And then out at Santa Cruz Island, there were no fish that were born before 1999. And 1999 was a very strong La Nina year, and a lot of blue rockfish settled on Santa Cruz Island, settled out even further in the Southern California Bight, Catalina Island and other places as well. And so it took a cold year for blue rockfish to recruit to the southern part of their range, whereas it took a warm year for blacksmith to recruit in Monterey. And then other fish were sort of tweeners. And this is the research done by Lisa Kriegsman here on kelp rockfish, which occurs commonly both in Central and Southern California. And her main finding, interesting finding, was that we made these collections in 2004 and 2005, and that she found in Monterey more of the fish had settled, more of the fish that we found belonged to year classes before 1999 during the warm phase of the Pacific Decadal Oscillation. Whereas in Southern California, most of the fish that we found were belonged to year classes from 1999 on. That is, maybe from the cold year and maybe the beginning of a cold phase of the Pacific Decadal Oscillation. And so she found, differing patterns depending on where you were in their geographical range. And so you have this picture of recruitment sort of sloshing around in California, depending perhaps on oceanographic conditions. But what is it exactly that makes it so that something like a damsel, a blacksmith, only appears have to recruit into Monterey during El Nino years? So one part of Kevin Walter's thesis was to look at the gonads, and what he found is that even in 1999, which was a very cool year, fish became reproductive. The males became reproductive, females grew ova, and he found size classes of ova both in Southern California fish and Monterey and Carmel Bay fish that are in the size range of eggs that are found on nests and so forth. So they're becoming reproductive in California even in a really cold year. So it really sort of brings up the question of what then is the limiting stage in the life cycle of a fish like a blacksmith. They're producing gametes. Is it fertilization, development of the embryo, development of larvae in the plankton, growth in the plankton, the way the currents affect their movements in the plankton? What is the limiting stage in the life cycle for these things? We don't know. And that when the young of the year do appear in, like blacksmith, in warm years, what is it? What leads to that? Is it successful local reproduction? That is, that they're making gametes and something happens in a warm year that allows the gametes to actually develop and go through their whole planktonic phase and appear on the bottom again? Or is it dispersal of larvae from Southern California? And I know that there's a lot of, sort of the word these days in fish ecology and marine ecology is, oh yeah, it's all localized recruitment. But it's not all localized recruitment, because in 1983, blue-banded gobies appeared in Monterey Bay, and during that El Nino. It was the strangest thing to see these little fish you hardly ever see outside of Catalina all over the Rocky Reefs. Those fish only live a year, isn't that right? There's no way there could have been a population hidden around somewhere in Monterey Bay that could have produced, just found great conditions for reproduction in 1983, put out a bunch of successful larvae. These fish had to have drifted up from Southern California. So the question remains, in these extremes of recruitment events, to what degree does successful local reproduction contribute to this recruitment versus drift of larvae from other parts in the range? We don't know. And then the differences between species, it's not just Southern California versus Central California. The species differ incrementally in their geographical distributions. I mean, there are big patterns, but even within a group, there can be incremental differences. Within Southern California, some species occur all the way out to the cold, San Miguel Island, some hardly even beyond Catalina, and so on. So it's incremental, and this just is an indication of that. These are the year classes of Senorita fish that Kevin Walters found in Monterey and Carmel Bay. Finding Senoritas, there's something about them that allows them to have successful recruitment every year. And my guess is, for them, is that they probably reproduce successfully around here, and their larvae are the ones that appear all these times. So what is the difference between these species? What is the difference between the Senorita and the others? And so I think there are a bunch of different questions. And if I was 22 years old again, if I was starting out, and if I knew what I knew now, which is an oxymoron, but I think that's something that I think would be really interesting to field that I think would be really interesting to follow up. So in what ways are species adapted to different latitudes? And it may be different for different things, that some may be maybe temperature, some it may be the effect of water movements on their late larval stages and so on. And in general, what are the roles of long-distance larval transport versus local reproduction of populations at the extremes of their ranges? And how does this change as populations wax and wane? This is, it's a practical question as well. In Southern California, when I was starting out in the 1960s and 1970s, you had to shoo away the blue rockfish, and but they disappeared for 20 years, and now they're starting to appear again. So are they going to start their own population? Are they going to be reproducing successfully? And so if you're a fisheries manager, do you have to look at this population and say, hey, this is something we have to take care of itself, or are the larvae all washing down from north of Point Conception or from San Miguel and Santa Rosa Islands or something like that? How does that change? When does a population start to go from being totally dependent upon long-distance larval dispersal to being self-reliant on their own reproduction? And what does it take in environmental conditions to do that? And another question that's interesting to me, and it comes from Bob Cowan's dissertation. Bob was a master's student here, and he did his PhD at Scripps and studied the sheephead. And I don't know where this hypothesis stands now, but he came up with the idea that really you can't just write off populations at the extremes of the ranges, because if you've got especially if you've got larval transport going different directions in different kinds of conditions, well, maybe these populations at the extremes are important in different years for helping to stabilize recruitment in the center of the range. I think that's an interesting question as well. And another question I think is interesting has to do with why localities can seem very different from each other. For example, Catalina Island, based on a lot of information I've seen, is really very much a warm water, what's the word I'm trying to think of? Warm water refuge. Not a refuge, but it's a warm water area. You find that's where we found butterfly fish in the 1990s and stuff. And blue rockfish, even in the early 1970s, when Ted Hobson was working out there, blue rockfish almost never appeared out there. So it's a warm water area, but 26 miles across the sea is the Palos Verdes Peninsula, which is a headland, little upwelling area, and there you have, it's a different kind of thing where you actually see much more evidence of cooler water species. And in the Monterey Peninsula itself, this is a regret, but the Monterey Peninsula itself, data that I've seen, gathered by a student of mine who never completed his thesis, Monterey seems to be a definite refuge, a definite enclave, that's the word I was trying to think of, for warm water species. And we did fish counts of Southern California expatriate species from Point Arguello up to like San Simeon, Piedras Blancas, and then from Point Sur and up to Monterey, and then a little bit up north too. And his data really indicate that. And it's one of those things that I have to urge him to finish before I get too much more senile myself. And so I guess, I don't know, these are all little things, but things that interested me, and I felt like I had to get off my chest. And the conclusions for anybody out there would be, don't be afraid to challenge authority. Don't be afraid to challenge the status quo. Even if you're talking to somebody who's just finished the study on this thing, and he tells you something, well, take it with a grain of salt. It may not be true. Don't be stupid about it. I mean, you're not going to undo force equals mass times acceleration squared. But don't be afraid to challenge authority. Try to plan ahead when you're doing research. Do good planning for your research projects and figure out what your sample size is going to have to be and all that sort of stuff. But then, if you find something interesting, don't drop it. That may be more interesting than what you found to begin with. And just stay excited about what you're doing. And since, I don't know if I mentioned this before, but I don't know if I'm ever going to give a scientific presentation again. I'm really retired. So I can't quit without, recognizing my mentors publicly one last time. John Stevens from Occidental College. I was mentioning with these guys today that I was in his comparative anatomy of the vertebrates class in winter of 1965. In that class, I looked up there and I said, that is what I want to do with my life, right there. And then he took me into his lab and made me a marine biologist, taught me how to dive, and started to change the course of my life. And then my major advisor at UCSB was Al Ebling, who's such a great scholar, and I hope I didn't disappoint him too much on that in that way. But he was a great advisor. He was wonderful at he had just dozens of graduate students, and he would do everything he could to keep things rolling. And he had a great group of students that I was working with at that time. And it was just one of the best times of my life there. And then one of the days that I remember there, this is in spring of 1969. This is right after the Santa Barbara oil spill, right after California Sea Grant got started. And when Al was getting sick of literally and figuratively going out on ships and studying mesopelagic fishing, and I got a call from him, and he says, hey, you and Bill Alavizian, come into my office, So we went in his office and said, hey, I just got a grant. You want to study kelp forest fishes? Well, yeah, and there it goes, and that's what I've been doing for the last 45 years. So anyhow, that's my talk for today. Thanks for listening to me.
So Rob, are you going to publish some of the stuff then?
You had to ask.
I did.
Well, yeah, I think, or hand it off to somebody to publish. What about you?
Yeah. It's funny that you should mention that, because today I was writing out notes for another paper I'm slogging through, and it was this old, spiral notebook, and it was creepy, but I opened it up and I was going through to see what pages were not written in, and the last page is all of your handwriting about that meeting that we had probably 3 or 4 years ago where we met in Davin. Hoping you wouldn't remember that. Yeah, where we sat down to talk about me publishing that. And so maybe it's all signed because there were your handwritten notes of what I needed to do to publish that.
In the first part of your the talk where you talked about the Capitola Reef, what do you think about the idea of just something novel? Right, these guys are sitting there; they don't move very much because nothing really changes. Suddenly, you dump this stuff; all these new smells are in the water, and they're just kind of like, "Whoa."
Yeah, that I've wondered about that actually. Whether because I was proceeding on the assumption that they had no way of knowing, no way of detecting that it was out there. I don't know if dumping several tons of concrete would have created noise that attracted their curiosity, or there was a novel odor, or if some fish gets out there and goes, "Hey, Joe," whatever. I don't know. It's strange credulity to me, but there's no other answer there's still no answer about how they could have gotten out there. So you wonder if there's some way that they could have perceived it. I don't know. It's an interesting question.
I wonder, I think it was Chris Lozo's lab where they took fish off the mainland and they're looking at the oil rigs and they move things around. These things they go across the entire Sunwater Channel, I think, right back to the reef that they came from.
Yeah. But they came from somewhere, but not going to somewhere that they didn't know about. But yeah, even so, that's where I think it might be worth having some telemetry studies in situations like that. Work it out. I have to talk to Chris a little about that. Yes.
On that same note, I mean, if they are sinks, right, and let's say it is smell or something new that attracts them to it, then they wouldn't really be sinks, right? Because they'd find that new place, and the ones that are in the Capitola Reef then wouldn't migrate over because there's nothing new going on. If that makes sense.
Yeah, I mean, they did keep migrating over.
So they just kept doing it for like a period of time.
Yeah, they did. There was an initial movement over. They didn't start tagging until after the reef had gone in. And the first time they visited the reef, there were fish on it,
Right.
and then they tagged the fish, and then they kept moving over. And I think Phoeniciensis may have the information about when they actually were caught.
Because they have some like artificial reefs that they made from subway cars in New York. Like I think they put them in New Jersey, and they saw a lot of success.
Yeah.
So if it's
They
true
do.
that they act as sinks, then that would really be success.
Yeah, I think it would depend. I was thinking about it just the other day, as a daydream while I was putting this together, that it really, whether it's actually a sink or not, depends on the amount of fishing effort that's going on, that's focused on that artificial reef, in comparison to what the fishing effort might have been in the natural reefs around there. And then it would be a sink, definitely a sink if the way I figured out, if the artificial reef actually attracted more fishermen as well.
Well, I think part of that artificial reef, source sink argument also depends on how old the reef is. I mean, it always seemed to me that it was a baloney argument. Because, maybe when you first put them in the water, especially if they're nice and rocky and not street carbon the rest of the way, they could probably attract fish, although, how they do it, why it differs from one study to the other, I don't know. But after a while, right, they're just another reef.
Yeah.
So after a while, they sort of probably are a source of settlement, everything else.
Well, that could be. It would be an interesting thing to sort of repeat that tagging and see, like now, and see if they still move out there.
Yeah.
Was the fishing pressure the same in those two places?
I'm sorry, can you say
Was
that?
the fishing pressure the same in those two reefs, the artificial versus the natural?
Well, there's several natural reefs, and I don't think. Well, maybe they did keep track of that. I don't know. You'd have to look at their theses. I think Al Solansky's thesis would have that in it, but I don't know the answer to that. It's a good question.
Because they might be following the bait and just going to their death.
Yeah, I thought about that too. Whether, I mean, fish are. Some of these fish are so opportunistic. Maybe at night they find some sardines or anchovies or squid or mysids or something like that, and they just kind of wander out and then here's snapping shrimp on the rock reef and move in. I don't know. Question.
I have a question about your evacuation rate study and whether or not you thought about bringing fish into the lab and just throwing them to grain and having an undergrad just watch them until they food.
Well, actually, the way that's what Todd Hopkins did. He did a lab study of it, and he did is excuse me, at the Steinhardt Aquarium, where he fed fish at a certain time, and then he pulled them out a certain amount of time later and did gastric lavage to see what was left. And yeah, I didn't tell the story about, for my master's thesis, I looked at stomach contents of black and yellow gopher, kelp, and copper rockfish. And I made some collections at night, in the morning. One year I did one at 6 in the morning, and another month I did one at 9 o'clock at night. And I was going to try and find out if they, what time of day they fed. That was not going to be sufficient anyhow. But at the end, after all of this was done, I decided in the lab to see, how long it takes for a fish to digest a crab. So I got a bunch of fish in the aquarium there in Marine Science Institute in Santa Barbara, and I put some crabs in, fed them to some black and yellow rockfish, and then said, okay, after 6 hours I'm going to check one fish. So I pulled one fish out of there and sacrificed it and opened up the stomach, and the crab
so you spent most of your career underwater in a wetsuit and scuba tank.
I spent most of my career at a chalkboard in front of a class.
Okay, that's true. Must be a research because you just go and scuba. And so in that time frame, other than the scuba equipment itself, how did your science change underwater?
Well,
did you ask different questions as you got more experience underwater? Did anything else change as far as how you did your work underwater?
Well, actually, I started off spearing fish and looking at stomach contents, and then I did an experimental study, field experiment study, which was at that time not really common. Not much of that had been done, and the tagging study that I did was pretty new. And then when I got to San Francisco State, I found that my options for research were sort of constrained by the amount of time I spent teaching. And so mostly I was trying to find projects that we could do, that we could get done. Like going out and spearing fish and bringing it back in the lab. Or maybe going out and doing transects somewhere where you didn't have to be out in the field making 40 or 50 dives a month for an extended period of time. And I think probably what I learned about the possibility of population genetics and what that might do, although I didn't learn how to do that. And some students got involved in that. That was new. And then when I worked with the National Marine Fisheries Service for 2.5 years in the late 1980s, they were doing a project on the midwater trawl surveys of pelagic juvenile rockfish. And that and some work I've done before when I was in New York, had to do with fish farming. I got more interested in pelagic stages of fish at that time. So new topics opened up, but I don't know if I changed the way I did science other than just figuring out how to address the problem that we decided to pursue. So I'm sorry. One thing that changed is I learned to take safety stops. Instead of just my old regulator weighing 200 pounds and going up, and unfortunately you get a couple more breaths on these old regulators when the pressure's less, and then popping up with the I shouldn't even tell you about that.
Remember those tanks coming back to the lab.
What?
We remember those tanks coming back to the lab.
Not very much. Yeah.
All right, well, let's thank Ralph.